Good afternoon. My name is Gwanghoon Jung from Busan National University, and today I will talk about improving genome variation curves for non-human sequencing data using machine learning. Genome variant calling is a task of identifying a single nucleotide polymorphisms and small insertions and deletions from genome sequence data relative to given reference sequences. Finding these SNPs and indels is a fundamental step for discovering biomarker genes associated with diseases. A lot of softwares for calling SNPs and indels have been developed. Deep variant developed by Google in 2017 is a highly effective deep learning model based on convolutional neural networks. Since deep variant has demonstrated exceptional performance in accurately identifying variants, it is widely used. To perform variant calling using deep variant, we follow four-step processes. First, we map the row sequencing reads to the reference sequence, while also marking duplicates and sorting the alignments. Second, we pre-process the alignments using tools such as GATK, employing techniques like in the realignment and base recalibration. And finally, we call the variance using deep variant. Although deep variant is quite successful for analyzing human sequencing data, its results for non-human genomes are not good as humans. The quality of non-human reference genome is often lower than extensively studied human genomes. Due to limited sample availability for non-human genome datasets, applying crucial processing steps such as in the realignment and base recalibrations becomes challenging. This means that conventional processing tools like GATK cannot be applied to non-human genomic data. That is why the performance of variation curves results from deep variant non-human genome is not as reliable as human data. To overcome this issue, we propose a machine learning approach to filter out false positive variant curves from deep variant. This model is specifically designed to improve the accuracy and reliability of variant curves in non-human genomes. Since it is difficult to obtain comprehensive and validated true variant curves for non-human genomic, we simulate scenarios by skipping the pre-processing steps for genome alignments using well-known human genome sequencing data. We mainly focus on homozygous and heterozygous SNPs and indel variant curves. We use genome in a bottle dataset to train the model. The genome in the bottle consortium aims to establish exceptional resources such as reference standards and reference methods and reference data for genomic research. In the genome in a bottle dataset, HG002 has used to train the model, while HG001 served as the test dataset. The reference genome employed in this study is HG38, which represents the most up-to-date version of the human reference genome data. In our study, we utilized deep variant to obtain variant curves as VHF file format and performed sequencing data analysis using tools like SemTools, Picard, and BWA. We extracted 7 features from VCF file, including the quality score of variant curve and variant type and variance positional information to build the model for filtering out false positive variants. We label variant curves of deep variant to binary types. Match all right are regarded as true cases, and the rest as false positive curves to train our machine learning model. The table below shows the miscalling rates of HG001 and HG002, where the miscalling rate is defined as the proportion of cases where the variant call is not true. As we didn't implement the index realignment and base recalibration, we observed that the miscalling rate for index tends to be higher than the rate of SNPs, whether they are heterozygous or homozygous. In our study, we implemented 5 machine learning models to filter out false positive variant calls, logistic regressions, and multilayer perceptron, tap transformer, random forest, and XGBoost. Among these approaches, multilayer perceptron demonstrated the highest performance, particularly in the context of heterozygous SNP and homozygous SNP variant calling. And this suggests that multilayer perceptron is effective in improving the accuracy of variant calling by reducing false positive calls. In summary, deep learning-based variant calling methods like deep variant face limitations when applied to non-human genome sequences due to the lack of information obtained from data pre-processing. To address this challenge, we have proposed a machine learning approach aimed at filtering out false positive variant calls. Our approach enhances the performance of deep variant specifically for non-human non-memory genome sequences. Okay, my presentation is ended here. Any questions?
We have plenty of time for questions, so please.
Thanks. Sorry if I missed it, but what is the proportion of false positives that were filtered after?
You mean the proportion of miscalling rate, right?
I suppose it's inaccuracy and precision.
Yes. Actually, this is the machine learning model's results and reduce of.
What question is the recall? Is the proportion 97?
Yes.
Is the recall of true variants, or I don't get what is the false positive?
The false positive means I should prepare the confusion matrix table, but the variant calls that is not match our right cases, such as if SNPs has happened, we the matrix confusion matrix contains SNPs that is actually right and SNPs variant calls that is actually correct, and variant call is correct, but it has different variant calls, such as SNPs has happened, but index has predicted cases. So the confusion matrix is made of the 4 components.
Yeah, to draw the confusion.
Thank you.